Now I would like to show you an animation. Hopefully, a lot of things that we have talked about will become clear when you watch this animation. Here is a bicep muscle. We are going to look at it at much higher magnification. So this is a biceps. This person was flexing the tendons at the end. In the middle, we will see individual cells. So this is a muscle cell bundle, a fascicle. This is individual muscle cell or myofibril. And inside this muscle cell, as we have said, that we have these myofibrils. They are enclosed in the cell membrane. The cell membrane is around these myofibrils. And now we are going to look at the structure of these myofibrils. But also I would like to point out that the endoplasmic reticulum in the muscle cell is referred to as sarcoplasmic reticulum. It has a very similar function to normal cellular endoplasmic reticulum, one of them being the storage house for the calcium ions. So let's move forward. These are T-tubules. This is the area where muscle innervates, a nerve innervates the muscle. So this is where the signal is delivered to the muscle cell to contract. So now we are going to see this structure in more higher magnification. So now you can see the striated look, the thick filaments and the thin filaments in blue, and thick and thin filaments. Now you can look at them both, giving muscle the striated look. Here we are going to look at the cross section, thick filaments. If you take a section to the thick filaments, you can see these thick filaments are organized also, irregularly spaced. If you take a cross section through thin filaments, you'll see these thin filaments are also organized they have regular intervals between them. So thick and thin filaments. This is sort of what we saw in that electron micrograph also. This is what they look like. Now, the zones that we talked about, I band, which is the zone where you just have thin filaments, and the H zone where you have which cover the entire length of the thick filaments. This is the process. The filaments are sliding. The thin filaments slide inwards towards the thick filaments. When thick filament, which are made up of myosin, they are activated, these filaments slide, resulting in muscle contraction. This is muscle relaxing. And the same process repeating itself, relaxing and contracting, when you flex your arm, this is what basically you're doing. You're making your actin myosin filaments slide against each other. So now let's see that I'm going to show you a higher magnification. Here is the muscle attached to the nerve, and here's the signal coming. Remember, this nerve, when it signals the muscle cell, it results in release of calcium ions from the sarcoplasmic reticulum. Once the calcium is released, it will interact with these molecules which are coating the actin filaments. They are preventing actin filaments from binding the myosin. So when calcium is released, the muscle is contracting, and when calcium is removed, the muscle relaxes, contract and relax. So muscle, as the calcium is released, contracting, and when calcium is absorbed, it is relaxing. So here you can look at the individual filaments, the thick filaments right here in the middle, and the blue ones, these are the thin filaments. Now we are going to, this is the sarcomere, is the zone between two Z lines. The M line is the middle of the thick filament. So these are the Z lines and the M lines that we are looking at, the A bands, the I bands, and also the H zone. So again, the process of muscle contracting. Now you can see how these individual filaments move relative to each other when muscle relaxes, and what happens when calcium is released from the sarcoplasmic reticulum, and these filaments slide against each other. Again, the calcium released causes the a contraction of muscle, sliding of these filaments relative to each other, the thick filaments and the thin filaments. Now we are going to look at individual filaments at molecular level. The calcium is released, it binds. Remember, we talked about this. Calcium released, it binds troponin, which is attached to tropomyosin, and a binding of this calcium to troponin causes a conformational change in troponin, which affects the conformation of tropomyosin, and the actin binding sites, myosin binding sites are revealed, and now myosin can bind actin, and it can use ATP energy to slide these filaments relative to each other. Here we have seen the process in a process of myosin pulling on actin filaments at a molecular detail in a different module. So this is basically how muscle is contracting. I hope this animation was helpful in understanding how muscles contract at a molecular level.